Hello there. Today I'm here to discuss with you the Bengaluru City University SEP syllabus, okay, for undergraduate program from 2024 and 2025 onwards, okay, which is framed according to the state education policy, that is SEP 2024 biotechnology syllabus I'm discussing with you. Okay, and this biotechnology course, which is a 3 years BSc course. Okay, so today I'm discussing with you the third semester paper. All right. So now let us go through the scheme of theory examination. That is pattern of BSc biotechnology theory question paper. It comes under SEP 2024. Duration 3 hours, maximum marks 80. First main, answer any 10 of the following out of 12. 2 questions. You are relaxed. So each answer carries 2 marks. So 20 marks here for 10 questions. Second main, answer any 6 of the following out of 8. Okay, 2 SQs will be there. So each question carries 5 marks. So this is 30 marks. Then question that is third main, answer any 3 of the following, that is out of 5. You need to write any 3, that is from 21 to 25. Each question carries 10 marks, that is total 30 marks. Okay, so you have questions from question number 1 to 25 in a question paper. So this is about the scheme of theory examination paper. Now let us go through the theory syllabus of third semester. The title is Biomolecules, paper 3 BTT 301. Okay, total contact hours 56 hours. In theory, you have 20 internal assessment marks. Okay. Now let us go through the syllabus. In this paper of biomolecules, you are studying unit 1, their introduction to carbohydrates and lipids. Okay, first you need to understand about introduction to molecular basis of life. Under that, once you study this molecular basis of life, then you should focus on carbohydrates. Here, you need to study introduction to carbohydrates, sources of carbohydrates, classification of carbohydrates, structure, properties, and functions of carbohydrates. Then about monosaccharides, that is isomerism, and ring structure, sugar derivatives. Then in oligosaccharides, sucrose and fructose, structure and functions. Polysaccharides in their classification as homo and heteropolysaccharides. Then homopolysaccharides, storage polysaccharides like starch, glycogen, their structure, reaction, properties you need to study. Further structural polysaccharides under this cellulose and chitin structure properties you need to study. The last content of the carbohydrates, heteropolysaccharides, glycoproteins and proteoglycans. So all these which comes under the carbohydrates. Next, under unit 1, you are focusing on lipids. Under this lipids, we need to study the classification functions of the lipids. As usual, introduction of the lipids, also study, then classification functions of lipids. Then properties. In this properties, you are focusing on saponification value, then iodine number, rancidity. Then hydrogenation of fats and oils, saturated and unsaturated fatty acids. Then further general structure and biological functions of phospholipids, sphingolipids, glycolipids, lipoproteins, prostaglandins, and cholesterol. So all these general structure and biological functions you must study. Then coming to the unit 2. Unit 2 is amino acids, proteins, vitamins, and nucleic acids. All right. So amino acids and proteins here, introduction to amino acids, classification of the amino acids, and structure of amino acids. Then concept related to the Zwitter ion and isoelectric point. Further, essential and non-essential amino acids, peptide and peptide and peptide bond. Then the new again, after this amino acids, you need to focus on the new concept related to the classification of proteins. Before classification of proteins, you must also study here introduction to proteins, then classification of proteins based on the structure and functions. Based on the structure and functions, you are going to categorize the proteins based on the structural organizations. Okay, then primary, secondary. Okay, in secondary, we have alpha helix, beta pleated sheets, then triple helix. You must study under secondary structure of the proteins, then tertiary and quaternary structures, then fibrous and globular proteins. Finally, denaturation and renaturation of the proteins you must study. Then under unit, second main B, that is vitamins, water and fat soluble vitamins, dietary sources and biological role of vitamins, deficiency, manifestation of vitamin A, B, C, D, E, and K. Then coming to the nucleic acids. Here you need to study introduction and discovery, then structure of purines and pyrimidines, nucleosides and nucleotides of RNA and DNA structures you need to study. Okay. So this is all about unit 2, that is amino acids, proteins, vitamins, and nucleic acid. Next, unit 3, that is enzymes and hormones. Okay, in enzymes, the first thing, introduction, then nomenclature and classification of the classification of enzymes. Then enzyme kinetics, factors influencing enzyme activity, activation energy, and transition state. Then enzyme activity in that specific activity. Further, coenzymes and their functions. One reaction involving FMN, FAD, NAD. Okay. Then metalloenzymes. Then next part, enzyme inhibition. Here you need to study about irreversible and reversible. Okay. Then under this category, you need to focus on competitive, non-competitive, uncompetitive inhibition with an example each. Then zymogens. Okay. Under this, trypsinogen, then chymotrypsinogen and pepsinogen, then isozymes, then LDH you must study. So this is all about your enzymes. Then second part under this unit 3, hormones. General introduction, protein hormones like insulin and growth hormones, steroid hormones like glucocorticoids, androgens, then estrogens, progesterone, and their basic functions, mechanism of steroid hormone action. So this is about unit 3, enzymes and hormones. The next one, last unit, that is bioanalytical tools and techniques. Here, you need to study about pH and buffer concept. Even you will perform this in the practical lab. So in theory also, you are going to understand this concept. Basics of pH and buffer concepts and chemical bonds stabilizing biomolecules like ionic bond, covalent bond, hydrogen bond, hydrophobic interactions. All these bonds you'll also study in proteins. Okay, there also you can recall all these bonds. Then chromatography, principle, procedure, and applications of paper chromatography and thin layer chromatography. You have other chromatography techniques, but here they are focused only on paper and thin layer chromatography. Then spectroscopy, under this principle, procedure, and applications of UV and usable spectrophotometry, mass spectroscopy, and atomic consumption spectroscopy. Last part of this unit 4 in your theory syllabus, that is electrophoresis. Under this category, you are studying principle, procedure, and applications of electrophoresis. Under this, agarose-gel electrophoresis and SDS page. SDS page is sodium dodecyl sulfate polyacrylamide gel electrophoresis. So this is all about your theory units. Okay, that is unit 1 you are studying in the beginning about the molecular basis of life. Then you must study about the carbohydrates, lipids, then amino acids, proteins, then vitamins, nucleic acids, enzymes, and hormones. Bioanalytical tools like pH and buffer, how to prepare the pH, like that, how to check the pH, all those things, and how to prepare the buffers. Then chromatography, two techniques, paper and thin. Then spectroscopy method, then electrophoresis. Okay, so you're studying this particular all these 4 units for 80 marks. Okay, if 80 for theory means 20 is your IA marks. Okay, assessment will be done, internal assessment for 20 marks. So this 20, how are we going to divide? House examination or maybe the test. Okay, that carries 10 marks. Written assignment, then presentation or project, term, 5 marks. Then class performance or participation in the class. So that carries 5 marks. So total 20 marks for your theory paper from the IA marks, from part of the IA marks. Then coming to the practical part, that is BTP 302 biomolecules. Okay, here you are writing theory, you are writing practical examination for 40 marks. Okay, 40 marks and 10 marks is IA marks. What are the experiment are you performing here? Preparation of buffers. Acetate buffer and phosphate buffer you must learn. Then definition and calculation of normality and molarity. First you need to understand what is normality, what is molarity. Then you need to understand the calculation part. One normal, one more, like that. Study of construction and working principle of colorimeter you must study. You need to understand this colorimeter instrument principle. Estimation of reducing sugars, that is glucose and maltose, by using DNS method. Then experiment number 5, estimation of protein by Biuret and Lowry's methods. Separation of amino acids or plant pigments by paper chromatography you need to perform. Extraction and partial estimation of protein from plant tissue by salting out method. The next one, estimation of alpha amylase activity by DNS method. So all these experiments you need to complete in your 3rd semester. As I told previously, you're writing this practical for 40 marks and 10 marks is your IA marks. This weightage of 10 marks we are dividing like house examination or maybe that is mock test carries 5 marks. Then class performance or participation during the practical time, that carries 5 marks. So total 10 marks. Now let us understand the practical examination paper. Okay. So practical examination paper which carries 40 marks, then duration 3 hours. Question number 1, estimate the amount of glucose in the given sample by DNS method and report. Or estimate the amount of protein in the given sample by Biuret method and report. So this carries 15 marks. If this carries 15 marks, you need to write the principle, then procedure, tabular column itself is a procedure. Then you need to plot the graph and you need to show to the report to the examiner. So all those includes 15 marks. Then question number 2, perform a paper chromatography experiment to separate the amino acids or pigments from the given material. Here you need to write the same thing, write the principle and procedure involved in those steps. This carries 12 marks. So in every experiment, the principle, procedure, and then result is compulsory. Okay. Then prepare a buffer of a given pH. You're studying in your syllabus, acetate and the phosphate buffer, right? So whatever the acetate and the phosphate buffer will tell those that particular acetate and phosphate buffer pH you need to prepare. I need to show to the examiner that particular pH. Okay, we may say some buffers, acetate or phosphate buffer, that buffer you need to calculate and you need to prepare. Okay, so that is about the preparation of the buffer of given pH. Then the 4th experiment, write the principle of salting out method or colorimetry. So examiner will choose whether you need to write about salting out method, colorimetry. So this carries 4 marks. Again, you need to write here the principle behind the salting out method and colorimetry. Then the last one, question number 5, that is class record, which carries 5 marks. Okay, total maximum marks 40. So you can take a screenshot of this one and you can keep. So this is all about your practical examination paper. I hope I made myself clear to you all, right? So if you are willing to purchase any book or if you want to refer means, you can go through these few books, okay, for as your reference. Okay. So these are some of the reference books. If you are willing, you can purchase. Okay. So this is all about your third semester theory paper and practical paper. Okay. Please make a note of this particular practical experiment and scheme in your observation book. Okay. In your practical observation book, you need to write these list of experiments. Okay. After the list of experiment, kindly note down this particular scheme of the practical examination. And you beginning only, if you understand the scheme of examination, it's better so that you can, during your regular practicals, you can focus on the important questions. Okay. I hope you understood whatever I explained here about your both theory and your practical regular classes and also the practical scheme of examination paper. Thank you everyone for watching this. I, Dr. Pratibha K, signing out for the day. Stay tuned with me. Thank you.